Thank you so much. Oh, recording in progress. Okay. Yeah, no, thank you. That was an exceptionally kind introduction. Joaquin and I overlapped a little bit in Cambridge, so we know each other from back then. So it's really nice to see familiar faces and to meet all of you. So thank you so much for turning up. As Joaquin said, I only started my lab, I think, a month and a half ago now. So it's early days, and obviously with COVID and stuff, we've been pushed back in terms of research. But I wanted to share with you a little bit about what we plan to do and some of the stuff that we've been working on so far. So I hope that it'll be enjoyable. So let me just share my screen. Share. Thumbs up if you can see that. Is that okay? Perfect. So yes, what I'm going to tell you a little bit about today, as Joaquin introduced briefly, are these in vitro model that we use called human gastruloids, which are effectively a in vitro model for anterior-posterior axial patterning in early embryo development. So the kind of primary question that we're interested in really is how we come to be, right? So how humans develop, how this embryo, or in this case, this is a fetus, comes to have this complexity and this organization. And I love this image from Lennart Nilsson back in 1960s. He sort of compiled all of these beautiful photographs of the early human embryo. And I think they really kind of took the world by storm, actually. I think people hadn't necessarily seen the human fetus in this much detail and this much complexity. And really it captures for me that kind of curiosity of where do we come from and how do we arise, and how is that whole process coordinated. And in particular, I think one of the most interesting questions in development is how do you get that complexity in a really reproducible way, but also from a starting population that is just a handful of cells. So this is an early human embryo, and you can see these extraembryonic tissues in blue around the outside of this sphere. And on the inside, there are these yellow cells that are going to form the actual embryo itself, the embryo proper. So the actual fetus will come from those yellow cells, and some of these extraembryonic tissues that are this kind of cloud of fluff in this image will come from those extraembryonic cells. And so really the question for me has always been, I guess, how does this happen? How do each of these cells make the decisions that they have to make, because they have to form all of the cell types of the body, whether that's a brain cell or a muscle cell or a skin cell, but they have to do that in a really coordinated way. And so it's no good one cell making a decision by itself. The embryo has to form functional tissues and organs, and it has to form a viable fetus at the end of it. And so the cells have to coordinate how they make those decisions. And I think that that's an area of our understanding that is maybe lacking at the moment. Maybe we don't completely understand how that coordination happens. And so the idea and the kind of curiosity really in human development is not a new one. People have been thinking for a long time about how we might arise, how we might come to be. So I just picked out a few really nice examples here from Nick Hopwood. And if you're interested, he's got an online collection of images called Making Visible Embryos. And so this was a really early example back in the 16th century where they thought about how human embryo development might happen. And you can see it looks a bit like a chicken egg, where there's this kind of growth happening, and you can kind of see over time there's an idea of vascularization, and eventually in this figure you can see kind of the outline of a fetus in this drawing. And so people kind of thought that this kind of materialization of the human embryo was kind of maybe a gradual thing, but it was kind of like a growth kind of aspect. And then obviously this is another famous example of how people thought human development happened, which is Hertzsprunger's homunculus, where he thought that the way that embryo development happened was that you had a sperm cell, and inside that sperm cell was a tiny miniature little human being kind of curled up tiny in the head of the sperm, and that all the egg was a kind of fertile garden, if you want, for which that tiny little human being could grow. And so the idea was that there was this predetermination inside the sperm of this form already kind of scrunched up inside the sperm that just then had to get bigger in development. And of course, another really famous example is Leonardo da Vinci's drawings of the fetus, which are obviously a lot more accurate than those ones. But I think that just captures the idea that, you know, we've always been interested in where we come from. And we've always had questions about how development and embryo development might proceed. Is it a gradual thing? Is it something where there's predetermination and then it's just kind of growth? Or how does that whole process happen? And it wasn't really until the early 19th century that we started to actually get to grips with what was really happening in the early human embryo. And so these are embryos from the Carnegie collection in America, gathered in the early 1900s from a variety of different sources, some of them ethically dubious. But they are effectively abortus material that was collected by doctors and gynecologists in America as a way of kind of cataloguing human development. And so on the top left is the earliest of these embryos. This is about a 20-day-old embryo, and in the bottom right is the latest in this series, which is a few weeks old. But the whole series covers most of development, most of gestation. And what was really important about these kinds of collections was that they had a huge amount of scale. There are literally thousands of embryos in these collections. The two biggest are the Carnegie collection in the USA and the Kyoto collection in Japan. But what they really allowed researchers to do was to kind of classify these embryos into stages. And so I guess the big advance here was that we could now say, okay, this embryo here, once it has this morphology, is a Carnegie stage 8 embryo, and then this one is a Carnegie stage 9 and 10, and so on and so forth. And so really that was a glimpse into early human development in a kind of static, characterizing, kind of classification almost kind of way. So just to refresh your memory about this time period known as gastrulation, because it's a slightly horrible word and I'll use it a lot, so I want to just make sure that we're all on the same page about what that means. So this goes back really to the original question that I posed, which is how do you go from a small clump of cells to something that has a body plan, a form, something that looks like a human embryo, a human fetus? And so what starts off as this small handful of cells, this group of cells known as the inner cell mass, which is here in blue, eventually in the human becomes a bilaminar disc, so 2-cell like thick disc where only this epiblast population of cells, so this blue columnist layer of cells here marked as the epiblast, that's the only embryo proper, and the rest of it is kind of extra embryonic tissue, as we thought about a little bit earlier. And so this bilaminar disc in the human embryo, surrounded by what's often called the double bubble of extra embryonic tissues, eventually undergoes this process of gastrulation, by which it forms a primitive streak that the cells of this ectoderm or the epiblast region move into that primitive streak, ingress in and around, and form the various mesoderm types of the embryo. And so effectively, this whole process of gastrulation, of forming a primitive streak in this epiblast layer of cells, this disc-shaped epiblast, is effectively doing 3 things. The first really important thing is that it forms the 3 germ layers. So this is kind of the canonical definition of gastrulation, that you form the ectoderm, which will go on to give rise to the kind of outside of the embryo, so the skin, but also the nervous system and other tissue types, the mesoderm, which will go on to give rise to the muscle and bone, and then the endoderm, which gives rise to a lot of the kind of internal organs like the gut. And so that's one of the really, probably the most key feature of gastrulation in any animal, and that's how this happens in the human embryo. The other really key feature of gastrulation is the definition of the axes. So one of the important axes that is determined by the embryo at gastrulation is not only this 3 germ layers, which is effectively the dorsal ventral or back to belly axis, but also the anterior posterior axis. So the primitive streak marks just the posterior part of the embryo. And so then obviously the side opposing that is the anterior end of the embryo. And so the origin of the primitive streak really marks that distinction between anterior and posterior, as well as dorsal ventral. And because it's a streak, also medial lateral as well. And finally, something that I think has been less appreciated really, but is definitely true of mouse development and human development is that at the point of gastrulation, there is a huge change in morphology in the embryo. And so this early epiblast, when it's just this disc shape, is actually kind of circular, it's kind of rounded in shape. And the only kind of distinction you can see on this Carnegie stage 7 embryo, so this is about 14 days old, is this slight line down the center here, which is the primitive streak itself. And so that disc, which is kind of circular shape, as gastrulation proceeds, will eventually kind of elongate out through, we presume, convergent extension type mechanisms to get this kind of elongated morphology, where the anterior end, you can see the head folds at the top end of that Carnegie stage 8 embryo, and then the tail bud in the most posterior end. And so all of these features have to happen at the point of gastrulation. And this is, I guess, what we want to learn a little bit more about. But I've been showing you kind of static pictures of gastrulation, and gastrulation by definition is a process. It's a really dynamic thing. And we're only kind of just getting a glimpse really of how dynamic it is, even in the mouse system, with some of the new technologies that have been kind of coming about and been gaining traction. One of them is obviously single cell sequencing technologies, and this paper came out a couple of years ago now, looking at the mouse embryo at the point of gastrulation and how the different cell types at the point of gastrulation go from just a few cell types to this kind of explosion in different cell types as gastrulation proceeds. And so you start off in this kind of left-hand side with kind of just epiblast type cells, and then over time you get more and more diverse cell types as differentiation goes on. But that's in time, and there's also very dynamic changes happening in shape. And so some advances in light sheet imaging, this is from Kate McDowell and her team now in Cambridge, where they took a mouse embryo at gastrulation and imaged individual cells throughout this time course. And they've plotted here the cells themselves and then colored them by their eventual cell fate. And so you can see over time that these cells are kind of rearranging, they're forming these beautiful patterns, but also the embryo is growing enormously. So this, at the top of this panel is the ventral view, so looking from the bottom at the top of the embryo, and then the bottom panel is the side view of the embryo. And you could see that it was kind of getting longer, but also these cells are rearranging relative to one another. And so gastrulation really has to be understood as a kind of dynamic process, not a kind of static snapshot of a kind of structure. And that leaves us in a bit of a conundrum when it comes to the human system, because we don't really have access to the human embryo at that time point. And the reason is that these collections of embryos that I showed you earlier, so the Carnegie collection and the Kyoto collection, really only start quite late in development. By late, I mean about 4 weeks. That's mostly for kind of technical reasons, that most women don't even know they're pregnant until about 4 weeks. And so there's not a lot of material available. And even if there was material available, it's quite hard to kind of find and to access. And so samples are much, much rarer at early stages from these sorts of collections. And in terms of kind of dynamic studies, in the UK at least, you're allowed to generate human embryos for research purposes, so from IVF, or use sort of surplus IVF embryos up until day 14. And so the law in the UK, in fact, it's a criminal offence to go beyond day 14, means that you have to stop experiments at this time point. And that it happens to correspond that day 14 is exactly the time at which the primitive streak is forming in the human embryo, and therefore gastrulation is starting. And in fact, the reason that the day 14 rule, as it's now called, came into being was precisely to stop embryos being cultured at gastrulation time points, so beyond day 14. Because before that time point, and we can discuss this later if you're interested, before that time point, you can take an embryo, mouse embryo, for example, and split it into two, and it'll give rise to two fully formed viable offspring. So some twins happen by that kind of means, where the embryo splits early on in development. And so you end up with two individuals. And so it was argued that actually before that stage, before day 14, the embryo hasn't become an individual, and after that point the embryo is an individual because it's got the anterior posterior axis, and if you split it, it wouldn't give rise to two separate individuals. Anyway, there's some call to change that, for the time being, at least, it's illegal to culture human embryos beyond 14 days of development. And so that means that there's a kind of gap really in our knowledge where we can't study dynamic human embryos after 2 weeks, and we don't have a lot of samples from these extant collections before 4 weeks. And so that gap in the kind of timeline of human development is sometimes known as the black box of human development. And so really, our work is kind of trying to plug that gap to develop in vitro models that allow us to kind of study this time point in a really dynamic way. Right, so that was a lot of preamble introduction. I'll get on to actually what we've been working on. And so, as Joaquin said, I mean, the majority of this work originated in Alfonso Martinez-Arias's lab in the University of Cambridge. And we started inevitably with the mouse as a system. I mean, many developmental biologists obviously use the mouse as a really useful model system, particularly because the embryonic stem cells of the mouse are really well characterized, and we know a lot about how to grow them, how to culture them, and how to differentiate them. And so a lot of the kind of work really was born out of this idea that researchers were growing embryonic stem cells from mice in a slightly artificial way, which is on flat plastic dishes in 2 dimensions and hitting them with a cocktail of chemicals that push them towards a specific lineage. So this kind of directed differentiation approach. And of course, that's not how the embryo works. The embryo doesn't have a flat plastic dish with a single chemical compound that pushes it towards one. The embryo has to balance the different cell types, and they have to allow communication between cells. And so I think conceptually, the idea was really, could we culture these cells in 3 dimensions, enabling these cells to talk to one another and to set up their own, signaling gradients or whatever that they do in the embryo in order to get a kind of self-organizing 3-dimensional in vitro system. And so the kind of methodology really is that we take pluripotent stem cells, either mouse embryonic stem cells or mouse iPS cells, induced pluripotent stem cells, and we dissociate them into single cells. Then a specific number of single cells are plated into each well of a 96-well plate. It's typically between 200 to 500, 600 cells. Routinely, we use about 400. And that is a lot smaller than an embryoid body, if that's something people are aware of, that people had been culturing these enormous, great, big, large spherical aggregates of embryonic stem cells and differentiating them. And they would find that they would differentiate, but in a kind of random, haphazard, kind of disorganized way. So this is a lot fewer cells. We're talking hundreds rather than thousands in a really defined condition in these 96-well plates. Now, these are round bottom 96-well plates, which means that the cell suspension eventually falls down to the bottom of these sort of rounded bottoms and coalesces into an aggregate. And so you can watch this process happen. These are mouse embryonic stem cells that are kind of coming together at the bottom of the dish, and they're finding one another just by kind of gravity, that they're in this rounded bottom dish, and they eventually stick together and form a spherical aggregate. And what was really exciting to us was that if we watch these spherical aggregates over time, they would eventually start to express some of the genes that are associated with gastrulation, but in an organized way. And so this is a brachyury GFP, sorry, I've lost the label here. And you can see that it starts to become expressed, but it's also only on one side. So it's become polarized in its expression. And that's spontaneous. The aggregate just breaks symmetry spontaneously and starts to express brachyury at one side. And for those who maybe aren't familiar with the field, brachyury is a really important marker of the primitive streak and therefore of gastrulation in the mouse embryo. And so if you leave these kind of ovoid shaped aggregates for longer, they'll start to actually elongate, so they'll change morphology. So this is a GATA6 reporter line, and you can see it's actually at the opposite side of brachyury, would be at the other end. And over time, they start to elongate and get much, much longer. And so they end up with actually an anterior and posterior axis. So we've done a lot of work really characterizing this system and whether it's a useful system and how similar it might be to the actual in vivo mouse embryo. So one of the things we've done is single cell RNA sequencing of these mouse gastruloids at late time points. So we go up to about 120 hours, so 4 days-ish, of culture of these in vitro systems, and we can look at the different cell types that are in our system at that time. And so we saw a number of different clusters, as one does with these sorts of single cell data sets. And we went back to the embryo and said, okay, which do these clusters match in the mouse embryo, sorry. One of the clusters was an NMP cluster, so a neural mesodermal progenitor, which are typically in the very posterior end of the mouse embryo. And they give rise to both the neural tube aspect of the axis, but also the kind of mesodermal compartment, so the somites and other axial mesoderm. And so these NMPs were next to two different clusters, one of which, cluster 6, was PSM-like, so paraxial presemitic mesoderm. Then a wavefront population of cells, a semitic population of cells, a differentiated somite population of cells. And on the other side of the NMPs was a kind of more neural population of cells, with actually even some head mesenchyme endothelial populations, and cluster 12 here is PGC-like cells. So really the gastruloid has a huge amount of diversity in terms of cell types that they sort of represent, which was really exciting to us, because that means that they've got a lot of complexity in terms of the differentiation that the cells do within a single gastruloid. But we also looked in time to see how these things were developing. And so a couple of years ago now, we took mouse embryos from early stages at about E6.5 and then followed them through development and just took the posterior part of the embryo. And I'll come back to why we did that in a minute, but effectively the mouse gastruloids don't have any anterior neural. So although they have neural tissues, that seems to be kind of posterior in origin rather than anterior. And when we did that, we could see that the gastruloids followed a very similar timeline to the mouse embryos. And so we could look at developmental time as it progressed in the gastruloids and as it progressed in the mouse embryo, and saw quite a good concordance actually between the two there. So as well as this kind of temporal dynamics of this addition of different differentiated cells, we also saw that there was a spatial component to the organization of these mouse gastruloids. So not only was there an anterior-posterior axis, which I've slightly touched on, where we have brachyury at the most posterior end, as well as CYP26A1, and a kind of gradient from the posterior to anterior. But there's also a dorsal ventral axis and a medial-lateral axis in the gastruloids. So they have a huge amount of spatial complexity in quite a robust and reproducible way. And we can compare directly the gastruloid expression to the embryo. And so, for example, this is an embryo on the left-hand side and a gastruloid on the right. And you can see brachyury in the most posterior tip of the gastruloid, as well as lunatic fringe in the middle part, and then PYY in the more endodermal component of the gastruloid system. It's not perfect by any means, but there does seem to be some concordance between the localization of gene expression in the embryo and in the gastruloid. And one of the things that surprised us most when we looked at this data set was that the Hox genes are actually expressed in gastruloids. So Hox genes, as I'm sure you all know, are really exciting in development because they have this collinearity where their organization on the genome is also correlated to the time that they're expressed, as well as the spatial organization of their expression. And that's consistent throughout the animal kingdom, so in Drosophila, but also in mouse and in human as well. And we were super excited to see that in the gastruloids over time, we could look at earlier gastruloids and later gastruloids and see these Hox cluster kind of opening up in time. So at early time points, we have the expression of some of the earlier Hox genes, and at later time points, we see some of the expression of later Hox genes. And as well as time, we also see some evidence of kind of nested spatial expression too. So some of the earlier Hox genes are expressed in a bigger portion of the AP axis than some of the later Hox genes like HOXD13, which is only in the very tip of the gastruloids. So that was super exciting to us. That implies that actually the mouse gastruloids can be used as a model to study gene regulation and some of these kind of developmental patterning type events as well. And so we were left with this idea that actually a gastruloid is kind of like a very abstract way of thinking about the embryonic body plan. So the kind of gene expression is in vaguely the right place relative to the embryo. We know that we didn't have any head, so we didn't have any brains particularly. So sort of post-occipital trunk development in particular. So the most anterior part of the gastruloid is the cardiac mesoderm. But the kind of gene expression organization is very, very similar in a very kind of abstract way. But the thing that was really confusing us was that actually, although the gene expression might be in vaguely the right place, the actual morphology of the embryo wasn't there. And so if we look at an embryo, for example, we have these gorgeous blocks of somites that are really important for forming the skeletal organization of the embryo. And so they form these epithelial blocks that you can see here marked by TCF15. And in the gastruloids, they're in the kind of middle region of the gastruloid, but there's no morphology there. And so those two things were really decoupled. And we wondered why there was that disconnect and how we could maybe introduce morphology into this system. And so collaborating with a team in the Hubrecht Institute in the Netherlands, they looked specifically at this somatogenesis morphology. And in the mouse, you have these gorgeous stripes of this marker gene UNCS 4.1 that you can see here marked in blue. And they found that actually when you embed these mouse gastruloids in matrigel, which is obviously a kind of ECM-like component gel, but actually in quite a low percentage, matrigel, you can actually start to see stripes appearing in these gastruloids using the same marker. And in fact, when they looked in a little bit more detail at these structures, so this is HCR and hybridization chain reaction, they could actually see that these somite structures had not only the striping pattern, but also had rostral causal identity within an individual somite. So if I zoom in here, you can see that an individual somite block has got an anterior-posterior organization as well. And so for some reason, this matrigel is allowing the addition of morphogenesis in these structures. We don't completely know why. We don't know whether that's the ECM providing components that are needed for these systems to kind of generate morphology, or whether there's some sort of physical interaction where maybe the embedding itself is constraining the gastruloid in order to form morphology. But I think it shows that actually these sorts of in vitro systems are really useful in a kind of bottom-up way of understanding what development is using in order to generate some of these features like morphology, for example. Okay, and so I want to move on to the human development. And so we thought for a while that this system was super useful in the mouse, but what would be really exciting would be to move the whole kind of system to human gastruloids. And so we kept kind of hitting back at people saying, you won't be able to do it because a human embryo is so different from a mouse embryo. And part of the reasoning was, okay, a mouse embryo at this point of gastrulation is a cup shape. And so you can look at the mouse embryo as this epiblast here that you can see in a kind of cup-shaped orientation. And a human embryo, as we've already discussed, is a flat disc. And so people said, you know, how will you know that you've got a good gastruloid? Because it's such a different shape, so you might have something completely different. And I think to that I think we had a good answer, which was effectively that the mouse embryo and the human embryo are incredibly similar if you kind of take a little bit of a conceptual leap, which is what people have been doing for a long time, that they say, okay, imagine we took a mouse embryo and we opened it up, we flattened it out, what would it look like then? And in that case, you would end up with a primitive streak that is on the posterior side of the mouse embryo. And if you flattened it out, it would be on one end, and you would effectively have a flat disc with a primitive streak in the middle. And so that's pretty much exactly what the human embryo looks like at gastrulation. And so the reason we thought that we might be able to do a similar kind of gastruloid sort of in vitro model was because the gastruloids haven't got any of the extra embryonic tissue. And so they don't have that constraint of being kind of rolled up into a cup shape, because that's, you know, introduced by the extra embryonic tissue rather than anything specific to do with the mouse. And in fact, we know that the gene regulatory networks between the mouse and human at gastrulation are fairly similar. And so it wasn't completely unthinkable that we might be able to get something similar to a mouse gastruloid, but with human embryonic stem cells. And so this is maybe a little bit too much detail, but in case anyone's interested, the mouse gastruloid system is very simple. It looks like this. We culture mouse ES cells just in serum and lymph, and then we aggregate them and put them in Ento27, which is a basal neural medium. And the only thing we really do to them is add in some chiron, which promotes Wnt signaling at between 48 hours and 72 hours. The only thing we had to change to get this protocol to work with the human embryonic stem cells, and I'm taking a very long story and squidging it down to a simple slide, was effectively just to shift that chiron treatment much earlier. And part of the reason we think that is because human embryonic stem cells are a much later stage, sort of equivalency in terms of potential to mouse embryonic stem cells. So human embryonic stem cells are actually more like mouse epi-stem cells, so slightly later in development. And so I think what we've basically found is that you have to kind of anticipate that chiron pretreatment much earlier with the human embryonic stem cells, and then they form human gastruloids, which I'm going to tell you a little bit about. So this is what human gastruloids look like when they're being made. So these are human embryonic stem cells that are coming together to form a spherical aggregate. This is a reporter line very kindly provided by Ali Bravanu's lab, where green is SOX2, blue is brachyury, and red is SOX17. Sorry, I should have put a slide here. And you'll see that they start to elongate similar to the mouse, and they start to polarize their gene expression as well. So over time, they'll go from this spherical to something kind of egg-shaped to something elongated in a very similar way to the mouse gastruloids do, with this polarized gene expression. And so we found that actually these structures have a really nice polarization. They have an anterior and a posterior end. They elongate really well. One of the things that we found that we were actually really glad about is that these human gastruloids don't have any evidence of any anterior neuron, so the same as in the mouse gastruloid system. And they also don't have any extra embryonic tissue. So that's a really important feature of the gastruloid system, because it means that it's not a complete embryo. So what we're not making, and I had to be very careful with the press here, was that we are not making little mini embryos in a dish. This is a model system that is a kind of abstract representation of part of an embryo, and it would never have any viability. It couldn't implant into the mother because it doesn't have extra embryonic tissues, and it hasn't got any anterior neural structures. So in fact, that's actually been incredibly useful for us because it's a simple system that we can use routinely in the lab. And so we characterize these a little bit further using a technique known as tomosequencing, which you might have come across, which is effectively where we took a gastruloid and we embedded it so that we knew which end was the anterior end and which end was the posterior end. And then this is in collaboration with our colleagues at the Hubrecht Institute. They sectioned tiny sections of the structure from anterior to posterior, and each section they subjected to RNA sequencing. So what that allowed us to do was to build up a kind of spatial map of the gastruloid from anterior to posterior in terms of gene expression. And so this is 5 mouse gastruloids that we looked at. So separate gastruloids for each heat map. And there's anterior to posterior along this x-axis. And you can see the kind of reproducibility from one gastruloid to another. So some of the genes that are expressed posteriorly in one gastruloid are also expressed posteriorly in the others. And the genes that express anteriorly in some are expressed anteriorly in the others. So that's been a really useful tool for us. And in terms of the human gastruloids, we've been looking very carefully at which genes are expressed where. I was super surprised when we saw this originally, that there are genes expressed along this whole axis of anterior to posterior in these human gastruloid systems. And so we're starting to kind of figure out what all these genes are, what they're doing, and why they might be organized in this way. But one of the things we were really excited to see is that there is this kind of peak of somatogenesis genes in the middle of the gastruloids. That implies that actually maybe we've got this continuum from the posterior through towards slightly more anterior somites. So we could look, for example, at signals and look at ligands and signal receptors and see where they're localized in these structures. So we think that we have kind of Wnt signaling in the posterior end and more BMP signaling in the anterior end. And so we're digging a little bit now into what that might mean, how that might arise, and how that might be coordinating cell fate decisions. So one of the other things we're really excited to do is to think about what might be human specific about human gastruloids versus mouse gastruloids. And so this method picked out a whole load of things that were the same between a mouse gastruloid and a human gastruloid. There's a lot of genes that are expressed posteriorly in both, for example. But there are also some genes that were different in the two species, and we're now starting to think about what that might mean and whether we can learn anything new about humans relative to mouse. So could we look at the gene regulatory networks in mouse gastruloids compared to human gastruloids and see, are there differences in how then these networks are configured, either in terms of actual genes that are expressed or their timings or their relationships. And so I mentioned very briefly this idea of somatogenesis. And so we saw, as I mentioned briefly, that there is this anteroposterior organization of gene expression in the human gastruloids, where we have sort of tailbud-like gene expression, followed by a kind of determination front gene expression, and then actually somatic gene expression. And so this is unpublished, but this is some of our human gastruloids developing over time. And you can kind of see, I mean, maybe it's a little bit tricky, but you can kind of see some segmentation appearing in time. So you can start to see these segments kind of following on in time, and we're starting to characterize this process a little bit further. And can we induce actual epithelialization and really characterize these structures? But I think the beauty of this observation was really that it allowed us to put a pinpoint on that kind of developmental timeline and say, okay, where are we in human development? And the appearance of somites is obviously a morphological feature. So we can go back to those collections like the Carnegie collection and say, okay, when do we start to see somites in human embryos appearing? And in fact, it turns out that it's between Carnegie stage 8 and 9, which is about 20 days of development. So this is actually really quite late. If that's where we're at with these human gastruloids, then I think that that's really exciting, because that's much later than we thought that we would get to, and also much later than the other models that exist at the moment. So where are we as a field? I mean, I think this field of embryo models is new, but it's also going to be gaining traction. So there's, I don't want to say the gastruloids are the only model that exists. I put this up here in case anyone's interested in some of the other models. There are some earlier blastoid models that have been in the press lately. There are these flat micropattern type colonies. And I think that they're all really useful in vitro systems to study different stages of development. And so this is a really nice review looking at the mouse embryo-like systems models that sort of exist already, and then the human ones, and how they kind of match up to the developmental timeline. So what's the use of these as a models or as a system? Well, I think that there are several, not only in terms of kind of ethical use. I mean, the mouse gastruloid system is a great way of reducing the number of mice that we need to use for experiments, which is obviously a real push in terms of policy, but also the kind of human system has potential biomedical applications. So in particular, things like screening for toxicology, toxicity of drugs for use in women of childbearing age, and kind of modeling diseases. And so that's kind of where we want to be moving a little bit more towards in the future. So just to give you a sneak peek of what we're kind of up to, we've been thinking about whether we can use gastruloid systems to look at what might be toxic to the embryo. So the embryo experiences certain compounds and chemicals that can affect development. So all-trans-retinoic acid is known to cause really severe defects. And you can see that in the mouse gastruloid systems, with increasing concentrations of this compound, they start to lose their morphology and they become much, much smaller. But also with the human gastruloids, they also change morphology and their gene expression changes. And one of the things we were super excited about is thalidomide, because I'm sure you will have heard of the really devastating effects of thalidomide that happened around kind of the mid-century, where women were given thalidomide as an anti-morning sickness drug. And it really sadly caused very debilitating conditions whereby the limbs were affected, miscarriages occurred, and it was a public health kind of outcry. And really the reason that that got through so much of the testing was because it didn't show any effect in mice at all. And so it was really a human specific response to this drug that wasn't predicted based on the animal models. And so we've been studying thalidomide with our gastruloid system, and in the mouse gastruloids, we don't see any kind of significant effect in the gastruloid development, but in the human ones, we do. So they don't have the same morphology and the gene expression is changed. So, I mean, it's early days. This is a very small scale screen, but I think it's really exciting. If we could begin to use in vitro systems like this to actually get at some of these questions before, you know, drugs go to market, I think that would be really powerful. The other thing that I want to start doing is thinking a little bit about modeling diseases. And so there are various conditions that are congenital abnormalities or birth defects in humans that affect the kind of organization of the body plan. So whether that's sort of skeletal abnormalities or axis elongation defects. So in this example, these are mice that have mutations that prevent the kind of formation of the posterior part of the embryo. And so we're going to be thinking a little bit more about how we can use gastruloid systems to model the kind of mechanisms underlying these conditions. And so really, I want to suggest that these gastruloids are useful both in kinds of developmental biology, fundamental development biology, but also towards slightly more applied systems, so disease modeling kind of approaches. So, okay, just to wrap up, I want to thank everybody, particularly Alfonso Martinez-Arias and his group at the University of Cambridge, from whom all of this work started, as well as our collaborators in the Netherlands, so Alexander van Oudenaarden and his team, and Switzerland, Matthias Luthoff and Denita Bull's team. As Joaquin said, I've just started up my new group at the Francis Crick, so we have some availability, some opening of positions, so do get in touch if you want to. I'm really happy to take questions and chat. Thank you.
All right, thank you, Naomi. That was fantastic. I enjoyed every second of it. Thank you also for giving such a paced introduction to the early development, because I mean, even for me as an entomologist who works with flies, it was very, very helpful.
Good, I'm glad.
So we generally take the questions live. So I will just whoever wants to ask something. Well, there's one in the no, that's it. Anna congratulating. So yeah, Ben Skinner of course has a question.
Fantastic work, Naomi, absolutely beautiful. So one thing that I want is how frequently do the gastruloids form when you just drop these aggregates of cells? It looked like they happen pretty much all the time.
Yeah, so we make them in 96-well plates. And in general, for a good experiment, and I'll qualify that in a minute, they almost all form, good single axis organized gene expression. So by almost all, I mean kind of 80, 90, 95%. What I mean by a good experiment is that the thing that's tricky is the starting state of the cells. So we're starting to realize that actually the density of the cells, the confluency of the cells, the colony size of the cells at the point of making gastruloids is very important, and any kind of deviation from that can lead to kind of all or nothing effects. So you tend to get either none of them form or almost the whole plate forms, and we're trying now to pick apart exactly how to kind of optimize that initial starting state of the cells. So yeah, I mean, I think that's partly a function of human ES cells anyway, because with the mouse system, it works pretty much every time. And I think that's because we know a lot about mouse pluripotency conditions, and we don't know all that much about human pluripotency conditions. So my feeling is it's the problem is our understanding of human pluripotency and kind of that sensitivity of the cell state. But yeah, I mean, it is a little bit tricky to get that right.
Fantastic. One thing that I wonder if you've been able to look at is how aneuploidies are treated in the initial stem cell populations. One of the big questions in the human IVF world is what happens with chimeric aneuploid blastocysts, and what's tolerated, what's not.
Right, it's such an interesting question, and no, we haven't done it, but yes, I really want to do it. So I think one of the really exciting things about this sort of system is that we can control the number of cells. So we can do cell competition assays. We can put one cell with aneuploidy versus 2 or 3 or 4 or 5 or 6. So yeah, I'm trying to get that off the ground because I think that would be really nice. I would imagine there's some sort of threshold beyond which the embryo can't kind of tolerate these sorts of defects. But there are certainly a whole load of conditions, perhaps not necessarily aneuploidies, but other genetic mutations where, patients exist with high amounts of mosaicism, and the feeling is that that's because they've got fairly low mosaicism, and anyone who happened to have higher mosaicism of that mutation just kind of didn't make it through gastrulation. And so I think that's a really interesting question, and it's something I want to do. I need more hands on deck to do all of these exciting things, but yeah, I want to.
Brilliant, thank you very much.
No worries.
Okay, Babis, go ahead.
Oh, that was a great talk. Thank you so much. So I was just wondering about the extracellular matrix, and if you have looked at all any integrin signaling which is shown actually to activate Winton notch and induce epithelialization in this system. Have you taken a look at it all?
Yeah, so in the humans, no, not yet. In the mouse, so another paper came out at it almost exactly the same time that our paper looking at the somite morphology came out from Jesse Wienvliet and his colleagues. And they found that actually by embedding these mouse gastruloid structures in matrigel, what was massively upregulated in the cells were the integrins and some of these kind of adhesion type things. So yes, I think that it's likely, because it's such a low sort of percentage matrigel, we're talking kind of 5%, 10%, it's not really a gel, it's not really kind of a gel that it's embedded in. It's just kind of free floating kind of ECM components. So I suspect that yes, that's the effect that the ECM is having, is just providing the kind of fibronectin that the integrins need in order to be able to kind of attach and start doing things. But we need to properly prove that by kind of, matrigel is this weird concoction of fairy dust, it's not really defined. And so I think we need to do that in a more defined way to be able to really say what it is that's causing that effect.
And regarding the somites, or somite-like structures that are formed, do these guys further differentiate? I mean, can you see any dermomyotome, any sclerotome compartments in there?
So yeah, so we've just got some single-cell RNA-seq data back from the mouse gastruloids at slightly later stages. And yes, there is some sclerotome and dermomyotome. Wow. It's not a lot. So I suspect that it might be a temporal thing. So it might be that, you have to keep them going a bit longer. We would love to be able to kind of take them and actually differentiate them properly towards certain things. And to be honest, I think that the benefit of these things is that they're so modular. You can kind of take them and push them towards whatever direction. So if you were interested in dermomyotome, they can easily be pushed towards that because the progenitors are there. So yeah, I mean, I think that will come.
Can I have one more question? Is that okay?
From my end, go for it.
So these guys, when they grow, do they have a polarized growth? I mean, what I'm trying to say is, can you actually see if you can locate any axial progenitor population within them? I know through gene expression analysis, you've seen these progenitors, the mesodermal progenitors, but can you see within this formation as they grow if indeed this population topologically seems to be like resident, so tail buds or chondroneural, chondroneural hinge kind of formation in there?
Yeah, so there definitely is a population of brachyury positive, SOX2 positive at the posterior end. Whether each cell is actually bipotent to give rise to both lineage, I think we would need fancier lineage tracing techniques in order to see that. We also really want to get very high mag kind of live imaging of actual cells moving, because we don't know completely how the axial extension is happening. We assume it's convergent extension because that would explain what's going on, but it's not necessarily the case. It could be, growth, it could be changing a cell shape. So yeah, on the to-do list.
All right, thank you. Fantastic talk, thank you.
Okay, Jacob, go ahead.
Hi, is this like flattened sphere-like structure of the human embryo something that's more unique to humans, or is it a primate thing? And if so, are there any animal models that could emulate that?
It's a good question. Yeah, so it turns out, or I think it's being understood, that actually it's the mouse that's the weird one. So yeah, the human embryo is this flat disc, but so is the pig, and the chicken is obviously a bit like that. There are various animal models like a lot of them are these flat discs, and actually the mouse is the rabbit, for example, is a flat disc. So the mouse is odd in being cup shaped, and I think that partly is to do with having, you know, litters where they just kind of need to squidge in as many embryos as they can into this uterus, which in the mouse is a kind of tube-like structure, or something to do specifically with rodent mouse development. So yeah, it's not specific to humans, the morphology of the epiblast, and people are definitely using obviously primate embryos to study this process, and also pigs in particular, because there are similarities in terms of morphology. It's not completely clear whether that similarity extends to kind of dynamics and also gene expression, gene regulatory networks as well. But in terms of morphology, there are lots of other species we can use to study that morphology.
I see. Thank you.
No problem.
So I'm going to ask myself some questions now. So this is sort of taking over from what Babis has asked and you have answered, because I was going that direction initially. So you've talked about having better live imaging resolution so that you can track cells, and that makes a lot of sense. But one of the things that have always fascinated me about this system is that before you started using Matrigel with the mice, by the way, I presume with humans you always do it with Matrigel, right? Or not? No, it's also okay in liquid culture. So during the regular gastrulation, you have aggression of the mesoderm, right, out of the epithelial layer, and then eventually they recover epithelial structure and make a coherent tissue. But there's a point where they are sort of free floating. So even beyond your convergent extension, how do the gastruloids that are growing and elongating in a liquid culture allow to preserve the integrity? Have you done anything in that direction, or is it something that still needs to wait for having or other techniques kicked into the cocktail?
No, it's super interesting, and I don't completely understand it either. No, so you're right. The gastroid is effectively, it doesn't go through an EMT because it was never epithelial to start with, right? So the embryo, mouse embryo, is epithelial, and then it EMTs to go through the primitive streak, whereas the gastroid is always kind of mesenchymal. And so all we see is the kind of epithelialization later, but it never which is the right way around, it never EMTs. So yeah, I mean, there is definitely a different cell shape on the outside of the gastruloid compared to the inside of the gastruloid. So our scanning electron microscope images of human gastruloids shows that the cells are really like flat around the outside, and they're kind of connected. So there is definitely I think that's what's holding the integrity, but I wouldn't say it's epithelial because, you know, they don't have polarity in the same sense. So it's a slightly tricky one. I'm not entirely sure how they do that or why they do that, but they clearly
that
do.
Sounds like a job for transmission electron microscopy of sections, right? That's
a job for somebody who's not a developmental biologist like me. That's somebody involved in cell biology, I think.
That's almost for a service. Yeah. So what about the cell adhesion molecule? So, I mean, in vitro, these cells, I guess the human cells express cadherin as well, right? Mm-hmm. So, do they maintain cadherin-based adhesion in culture?
Yeah,
in the gastruloid.
Yeah, so obviously the human ES cells express E-cadherin, and then once we hit them with chiron, which is actually, I didn't explain this properly, but it's in adherent culture that we hit them with the chiron before the aggregation, they actually go from E-cadherin to N-cadherin in 2D. So not completely switch, but they start to express N-cadherin, and then as they differentiate in the gastruloids, obviously they go towards N-cadherin for the fully differentiated cells. So yes, I think that they are.
Does this mirror what happens in the embryo?
Well, not in the human, obviously. I think in the mouse, it's known that the E to N cadherin switch happens at the point of gastrulation. So that's kind of to be expected, that they're on the cusp of the primitive streak kind of early differentiation. So yeah, I suspect that that's what's happening.
Okay, so if I can finish with something a little bit more provocative. So You have mentioned at the beginning this, you said in passing that so far the limit is 14 days for the embryo, and there's a very clear reason for that limit, which is not going to go away. So, but you just said it like if this was something possible, and you've also mentioned the idea of that you don't want to go anywhere near a gastruloid with extraembryonic tissues. Precisely for that reason, right? But when one starts designing the experiment of matrigel, and of course there are many variations of extracellular matrices mimics that you could use and experiment and see which ones get you further. But yeah, there's also the temptation of mixing this up with some trophectoderm cells that you can derive from in vitro. I mean, the technology for that has been there in petri dishes for a long time now. At least with mouse. I don't know so much for humans, but I presume it is easy to derive trophectoderm cells. So you could perhaps think about coating this with trophectoderm cells. Perhaps you could even put a suicide gene in them, so that they only last for a while. So do you think there's a scope for this technology? Is this an absolute serious no-no? I mean, and whether it is or it isn't, how further do you think you will ever close the gap and reach to the 4-week stage structure? Have limbuts?
Yeah, I mean, that's a mega question. Oh my goodness, I could answer for hours, but okay, I'll try and keep it brief. So yes, there is calls to extend the 14-day rule. In fact, the ISSCR guidelines came out yesterday, if you saw them, suggesting that on a case-by-case basis, human embryo research should be allowed to go beyond 14 days. Obviously, that's not been written into UK law yet, so we can't start doing that right this second. But if that did kind of hit the legislature, it's quite possible that sort of hard line might move. In terms of kind of experimentally adding extraembryonic tissues or indeed neural tissues, I mean, that's the question we always get. Could you get a mini brain and plonk it on the end of these things and create a complete axis? I mean, my answer is always the same, and I think it's partly for ethical reasons, but actually partly for scientific reasons. I'm not sure that it makes a lot of sense, because I think the beauty of these systems is that they're so simple that effectively we've taken this really complex structure like the mouse embryo, for example. We can culture mouse embryos, we can use them, but actually having a gastruloid model is really useful because we can decouple the extraembryonic tissue, the influence of the maternal environment, the influence of the brain on providing signals, and we take away all of that complexity and we have a really simple bottom-up approach where we say, what do we need to add to the system to get morphology, for example? What do we need to add to the system to get this cell type? And so actually the beauty of the system is in its simplicity. And if we start adding in loads of stuff, we get back to a really complicated state where it's hard to understand.
But the answer to the question may be trophectoderm cells.
Maybe, I think, and then we have to say, are we learning something from that? So if we were looking specifically at the interaction between the trophectoderm, then yeah, okay, maybe we would learn something from that. And then we have to ask, is it ethically justifiable? And that's a different question. And different people will give you different answers. So yeah, who knows? I think that has to be a public debate, right? I think as scientists, I can sit here and say, yes, there's good reason for looking at the human embryo at 3 weeks, or no, there's not good reason. But I think in the end, it comes down to what do the public think? What do they want? How much do they trust the scientists? The decision was taken really from a kind of middle ground point of view to try and balance what the scientists wanted to do after IVF and what the public thought was acceptable. And there was a lot of religious groups who said actually no work on embryos is at all acceptable. And so this was found as a kind of middle ground. And so I think we have to be quite careful if we're talking about extending that we are not evading public trust in science. Mm-hmm. But that's my personal point of view. So I don't know. We can all argue about that, I'm sure. Maybe over a drink sometime.
All right. Are there any other questions?
Could I just ask for a sort of point of information? Did I hear you correctly, that in the thalidomide disaster, it turns out that the phocomelia was really human specific? and didn't show up in any model systems. Did you say that? Or...
So if I said that, I misspoke. So yeah, so it doesn't show up.
No, that's fine. If that wasn't the situation, then I have no question. It's just that in a documentary, you know, totally unreliable source, but in a documentary about thalidomide, I remember hearing that when rat results were looked at retrospectively after everything had gone wrong, that limb malformation in fact had been found, but it wasn't reported. It was overlooked at the time of the toxicity testing.
Yeah, so the rat and the mouse was a little bit dubious. The rabbit does show effects with thalidomide. And so, you know, now the law is that you have to do a rodent and non-rodent test of teratogenicity. So you have to do, mostly people do a mouse and a rabbit or a rat and a rabbit, in order to try and get away from anything species specific like thalidomide that doesn't show up so well in mouse systems. So yeah, it's not human specific completely, but it was not shown in the mouse example.
Thanks very much. And absolutely super talk. Thank you.
I'm glad.
Well, I think with that we can conclude our session. Let's thank Naomi once more. It was really, really, I think, a crystal example of what an excellent talk is. Also, I think, very accessible to elegant researchers and to undergrads if they were here.
Good. I'm
All right.
glad.
Thanks again for taking up our invitation, Naomi.
No worries. Thank you, everyone.
Bye.
Bye.